When it comes to the concept of the central dogma proper, starting with the transcription, we are now actually going to use here different types of codes or jargons. If the DNA strand is running from 5' to 3' direction, that DNA strand is known as the sense strand, or also known as the coding strand. But if the DNA is running from 3' to 5' direction, it is now known as the template strand or the anti-sense. If the transcription will happen inside the nucleus, that part that would simply be created as a form of mRNA or messenger RNA, it will now be known as the coding mRNA. And there are actually two types of mRNA. We have the presence of a pre-mRNA and the presence of mature mRNA. And we're going to discuss this a little bit deeper later on, okay, as we discuss the concept of transcription. Now remember that the coding strand of the DNA is possessing the same orientation and direction as the coding strand of the mRNA. Coding strand or the sense is running from 5' to 3' direction. The coding strand of the mRNA, either pre-mRNA or matured mRNA, would always be 5' to 3', okay? We are going to read this from 5' to 3'. That would always be our coding strand for the mRNA, okay? So, as you can observe here, when it comes to the coding strand, on the DNA, there's the presence of the thymine. Then the template strand of the DNA, it is now possessing its complementary, which is adenine. Then when transcription will happen, that adenine would be complementary with uracil, okay? And if we're going to compare coding strand of the mRNA and the coding strand of the DNA, they are the same except for the presence of thymine for the DNA and uracil for the mRNA. So now, when it comes to the presence of the DNA to mRNA transcription, it is actually divided into three major steps. The presence of initiation or the start, elongation or the addition of the RNA nucleotides to create the mRNA, and the third one, it is the termination, which would trigger the end or the stoppage of the transcription process. How is the mRNA created? Well, there are actually different types of proteins that are responsible for this. But we're always going to start with the presence of the term as the promoter. The promoter, it is now the TATA box, okay? TATA box. It is known as the segment of the DNA in which the concentration of thymine adenine is very high. The transcription will always start on this location. Why on the TATA box? Since, if we're going to recall on your discussion on the previous module, the TATA box or the thymine-adenine connection, it is only possessing 2 hydrogen bonds. Unlike in the case of guanine and cytosine, which is possessing 3 hydrogen bonds. The 2 hydrogen bonds that are present in between the thymine and adenine, when it comes to the DNA strand, it can easily be broken down by the enzyme known as the RNA polymerase. Okay, the RNA polymerase, from the name itself, polymerase. Polymerase, it is the enzyme that is responsible for the creation of that certain type of polymer. If you can recall, on replication, DNA polymerase, because it is now creating polymers of DNA. RNA polymerase now on transcription, since it is now creating RNA polymers. Okay? So when it comes to the process of this, the initiation will actually start at the promoter region known as the TATA box or the continuous thymine-adenine connection. Then after the presence of the promoter, the RNA polymerase will actually enclose that DNA strand and the process of elongation will now start. Remember that the promoter region would be responsible for the start or the initiation of the replication process with the attachment of the different parts of this RNA polymerase. And now the RNA polymerase would now be responsible for the attachment of the complementary strand that is present on the template strand, once again, we are going to utilize the template strand when it comes to the transcription, okay? The template strand, then it will now attach here the presence of RNA polymers, okay? The RNA nucleotides, namely adenine, guanine, cytosine, and uracil. We are now going to utilize these materials. Then, it will now simply be attached from 5' to 3' direction, and as you can observe here, the unwinding will simply happen on the right side of the RNA polymerase, then on the left side is now the rewinding, or just like what is being done by the DNA gyrase. The RNA polymerase complex is actually doing all those things. Okay, the RNA polymerase is now responsible for the unwinding and even the attachment of the RNA nucleotides, which will now create here the pre-mRNA or the young mRNA. Now, when it comes to the presence of eukaryotic cells, it is actually quite complex. There is actually an example on the previous slide. There is actually an example of a prokaryotic cell transcription. While in this case, in the case of the eukaryotic cell transcription, there are additional types of transcription factors, since the DNA that is present on eukaryotes are definitely longer than the ones that are present on prokaryotes, okay? But basically, they will just create the transcription factors for initiation, then it will now be attached on the Tata box, then the RNA polymerase will now enter the picture and do the process. The same process that is done earlier. After the process of transcription, a primary transcript will be created. The primary transcript is actually created by the presence of the mRNA, then the presence of the methyl cap, and the 3' poly A tail. From the name itself, poly A tail, because there is now the presence of a lot of A's or adenine on the 3' end of that transcript. While on the 5' end, there is now the presence of the cap known as the guanosine triphosphate. Okay? Guanosine triphosphate that would now be on the 5' cap, while poly A tail that would now be present on the 3' end of the primary transcript. This primary transcript, it is once again known as the pre-mRNA. It is not the mRNA that we are going to utilize for the translation, since we need to do here the process known as splicing. Primary transcripts are actually modified to produce mRNAs, and the pre-mRNA splicing is accomplished by spliceosomes. Because when it comes to gene expression, all of our cells are actually possessing here the same set of DNA, except for our gametes, or except for our sex cells. When it comes to our somatic cells or our body cells, the cells that are present in our skin, the ones that are present on our gums, the ones that are present on our nose, on our muscles, they're actually possessing the same DNA. Same 23 pairs of diploid chromosomes. It is now possessing the same genome. But the problem is, why is that different types of proteins were created? Those different types of proteins were created because of the presence now of the spliceosomes. The spliceosomes will now be responsible for removing the unwanted part of the mRNA that would not be responsible for the creation of the specific protein needed during that specific part of the body. So basically, the pre-mRNA would simply be spliced, okay? It is spliced, not sliced. It will now be spliced to create the mature mRNA, and this mature mRNA will now be containing the usable codons, the 3-letter nitrogenous base words that would now create the specific proteins needed for the process of the gene expression. Okay? Basically, there are two parts of the mRNA, specifically the pre-mRNA. The pre-mRNA are possessing not only the caps, but also exons and introns. Exons, they are the coding part of the mRNA. The exons are possessing the specific instructions needed for the creation of the needed proteins, while the introns, they are the unusable or useless part of the mRNA. Basically, this is being done by the following process. The first one, it is now the presence of the spliceosomes utilizing now an RNA known as the snRNA. The snRNA, or the small nuclear RNA, they forms base pairs with the 5' end of intron and at branch site. Okay? So, basically, the snRNA will now attach on the part of the intron. The introns, they are colored pale, while the exons, or the coding part of the mRNA, they are colored purple. Okay? So, this is the intron part of the mRNA. Okay? The snRNA is now actually present inside the complex known as SNRPs or SNRPs. This is now the spliceosomes. Okay? The spliceosomes, they are now containing the snRNA that would now be directing which part of the mRNA strand is considered as the exon and which part is the intron. Obviously, they will now attach themselves on the introns and not on the exons. Then, the spliceosome complex would actually move to each other. Then, after they move to each other, they will now create a lariat or a shaped lariat that is just like a pretzel knot. Okay? This pretzel knot will actually be detached from the pre-mRNA. Then, the same spliceosome will now attach the one end of the exon to the other end of the exon, creating now the mature mRNA, while they will now splice or remove the intron. Okay? They will be removing the intron and they will just retain the exons. And the exons will now create the mature mRNA ready to be utilized for the codons that would be responsible for translation.